Hey, I think I can hear you. Can you hear me? Yeah. Hey, man. Yeah, you got me.
Perfect. What's up? Thank you for your patience.
Yeah, man. It's great to meet you.
So what's up?
Um, yeah, man. I just wanted to like kind of walk through some this immune bio. I mean, I think it's interesting, and you pointed out a lot of the kind of critical flaws with it. But that said, from like a mechanistic perspective, I think it just has, you know, maybe more than a 40 40% chance, closer to like 50-50 at this point,
Okay.
which is like not really reflected, I think, and it just makes the short a little scary and makes more sense as like a small long.
Okay.
Um, I can kind of walk you through why I think that. Um, I don't know if you have any kind of questions or you want to direct it anywhere, but
Not
happy
really.
to
I mean,
just
I'm
kind of
pretty
walk you through.
confident in my short. Um, I don't think it has a chance to succeed, but that's what makes a market. If we could both win at the same time, I would love that. But one of us is going to
Yeah,
be right, one of us is going to be wrong.
yeah, I mean, it's speculative, and like to your point with your short thesis, like these Alzheimer's trials fail, and this one's particularly short and small, and the predicate data isn't that strong. That said, like I think there's a lot of mechanistic rationale why this could work, and there's also some data from like the TNF alpha inhibitors that suggest this is preventative. And I think the one data point that is like most interesting to me is it's coming from this 2017 Nature article, coming from Ben Baer's lab. It's titled "Neurotoxic Reactive Astrocytes Are Induced by Activated Microglia." Essentially,
Okay, I'll pull it up.
that paper kind of mechanistically lays out potentially how it is that amyloid is causing neurotoxicity.
Wouldn't this
And
apply to MS?
no, because the MS is kind of like an autoimmune. It reacts to antibodies that are like mistargeted,
Well, T
right?
cells too.
Yeah, there's also T cells, but it's still like misappropriated immune targeting, right? Whereas this is just like um, amyloid beta. It shouldn't really be in the brain. Part of the process of sleep every night is, I don't know if you've heard of the glymphatic system.
Dude, I've been in biotech 25 years.
Yeah, so like, it's becoming more clear, I think mechanistically, at least to me, that the brain is supposed to clear its junk that it accumulates during the day from just normal processing and being awake. And the way it does that is by shrinking the neurons and then kind of pumping through like delta waves and beta waves, whatever, all of the junk out of the parenchyma and into the veins and the vessels, and it gets kind of flushed out. And the problem is when you don't have a good flushing process, that leads to accumulation, and then that accumulation eventually chokes out the organ and
What does
kills
this
it.
have to do with TNF?
So, so what that has to do with TNF is like a lot of the risk factors for developing Alzheimer's disease are either neuroinflammatory, as identified by GWAS, and also from an environmental
I
perspective
thought it was ApoE.
related.
I'm sorry if I'm cutting
Yeah,
you off.
no, ApoE is also kind of a neuroinflammatory
I think
lipid.
the ApoE is the main thing that's really the only thing that's kind of been
So yeah, if you look
valuable.
at like GWAS studies from like, there's one in Nature Genetics from 2014, and there's been several
I
others.
mean, you don't need to look at any
There,
studies, right? ApoE is the main thing people focus on from a genetics perspective. I mean, that is the cardinal, other than autosomal dominant, ApoE is it, right? I mean, that's the thing.
no, there's others, and if you were
Okay,
to,
so you're telling me that when you go to get a gene panel, ApoE is not the most important contributor to familial Alzheimer's.
so I'm saying it's very important, but I'm saying it's also part of this neuroinflammatory cascade. that's happening,
But in terms
right?
of the most important gene for familial Alzheimer's,
Yeah, it's the most important one, but the way it works is through this neuroinflammatory cascade. It's
you think
sort
so?
of it's inconsistent with what I'm saying. Yeah.
How do you
So
think of it? How do you think it's neuroinflammatory? What part of it do you think? What's its job?
so there's a paper that actually goes into the mechanism of this.
But it's is it a transcription factor? Is it a protein that does something specific? Pretty sure it has like a
No, I mean
very specific
it's a
function.
yeah, it's like a lipid, right? It's
Right.
found in like yeah. So I think it has to do with this stability of these like endothelial cells or the membranes as part of like lymphatic clearance. And so what I'm trying to get to, right? Let me just kind of finish what I was trying to say. The lymphatic system is meant to clear out the brain. If you don't have good clearance over the course of decades, you eventually accumulate all this junk and it kills the brain. There's a lot of risk factors with respect to neuroimmune genes like ApoE and BIN1 and IBO. There's a lot of these markers that have been kind of accumulating on microglia and astrocytic genes more than neuronal cells. And then the other risk factors involve like cardiovascular risk factors. So people who have diabetes, people who have statins. And so basically you have kind of a motivation to keep those vessels nice and clean and open. And so back to this mechanistic paper from 2017, basically this is coming out of Ben Baer's lab. And it's kind of a shame because Ben Baer's published this in 2017, kind of shaking up the world a little bit. And then he actually died that year from pancreatic cancer. And there hasn't been a lot of follow-up. There's been a few papers here and there, but it's kind of tough when you lose the leader in the space. But what he found is that these amyloid species kind of start to accumulate in the brain, and then that leads to activation of the microglia and then eventually of the astrocytes, and together they start to kill off the neurons. And he found, if you look at, I don't know if you have it open.
Yeah.
If you look at like figure 1A, there's like kind of two distinct types of astrocytes that he found, this like A1 and A2, and it's the A1s that are actually neurotoxic, right? And the whole paper kind of goes into the mechanistic drivers of these A1 cells and their killing capacity, and they're dependent on IL-1 alpha, TNF, and C1q. Whereas these other A2 non-killing astrocytes just don't have these expressed at all. And then kind of later on in the paper, they go on to prove that like just activating these three factors is necessary and sufficient to create kind of killing astrocytes. So I don't know, like maybe, X-Pro is just one of those three legs of the stool. It's maybe not enough. But I think it's certainly interesting. And then, going back to X-Pro and the design of the trial itself, this, I think one of the criticisms is rightfully so, is like, this is a 200 patient study over 6 months or so, which is like, basically lecanemab and Kisunla are like 2,000 in like 2 years. But actually, if you look closely at the data, lecanemab would have hit stat sig or been very close to it at 6 months.
With a lot of patients.
With 2,000 patients, that's for sure. But Immune had an interim analysis, right? And they looked at like the standard deviation, and they saw that it was consistent with their powering assumptions. So I think, at least that's behaving okay. And then the other thing is they're enriching their trial for patients who are thought to be kind of like super responders or super progressors. And so they have things like ApoE positivity, they have diabetics, they have elevated ESR, which is like an inflammatory biomarker, and elevated CRP. And you have to have like, one of these 5 markers, and like 20% of them have double, or at least 2 of them. And so like you have this kind of setup where you're accumulating patients that are probably on like a rapid progression because of just how sick they are from a cardiovascular perspective to Alzheimer's progression. At the same time, like with lecanemab was pretty close, or if not likely to hit stat sig at 6 months. And so like this drug, I think we know from the science paper, and I think you did a lot of good work on this, is definitely active. And so the question is not, is this a real drug? Is like, it's more like, does it work? And I think there's just enough tidbits here, including their own, like, albeit weak biomarker data to suggest this thing is definitely anti-inflammatory, and it potentially could be doing something, as evidenced by like some of the MRI scans where they see like more white matter remyelination, and the reductions in like NFL, neurofilament light protein, and as well as some of the other inflammatory markers. I think there is still a question of like, does it need to get into the brain? And on that point, like, I'm not sure that it matters that much. Maybe the little amount that does get into the brain should be enough. But if you look at like, the TNF alpha inhibitors, those therapies are able to like reduce the incidence of Alzheimer's in patients with RA, with rheumatoid arthritis, and they're not getting into the brain.
That's a lot of that's a little bit of a dubious statement, but
no, I mean there was a paper published in PLOS One a couple of years ago.
Was it a prospective clinical trial?
No,
Okay.
no, it was
Just
like
making sure I
basically
didn't miss
looking at
anything.
like it was basically looking at tens of thousands of just patient data from like EHR or whatever. Where basically you take a cohort.
You can't take the EHR data to the FDA and get approval, right?
No. What I'm saying is like there's evidence to suggest that these TNF antibodies are protective against Alzheimer's. And yeah, it's not high quality data. That's what I'm saying, is like this whole story is like standing up on like twigs and like
Well,
pieced together with like staples and bubble gum. But
yeah,
it is like it's definitely like enticing, and I think with like a 5-to-1 payout, I don't know that, like, that basically implies, I guess, like maybe 20% chance that it's successful. And I think it might be, I think it's a little better than that.